The topic name is mutation. So, in this chapter, we will learn mainly about mutation and its types. So, before starting with this topic, let's know the basic concepts regarding the mutation and the definition of the mutation. The mutation it is a sudden change in a genetic makeup of an individual or any hereditary change in the genetic makeup of an individual which is mainly caused by the simple recombination of the genes is called as mutation. So these mutation it may change the arrangement or structure of an DNA, or it may change in the number of an chromosomes. So in this way the mutation will happen in an individual. The term mutation is given by the scientist Hugo Duris. We already know the scientist Hugo Duris. He gave the mutation theory and he did experiment on the primrose plant, that is Ainothera lamarckiana. So he studied in detail in regarding this mutation in the union primrose plant and he gave explanation regarding the mutation in the individual. So when we see the mutation, we mainly study these mutation into a different types. One is based on where the mutation is happening. So the mutation which are occurring in an germ cells are called as germinal mutations. So the germinal mutations are those the mutations which are happening in a germinal cells. And another one, that is the somatic mutation. The somatic mutations where the mutation which are occurring in a body cells are a somatic cells. So there is a difference between these germinal mutation and the somatic mutation, where the germinal mutations they can pass these changes or mutations to the next generation, whereas the somatic mutations they doesn't play any role in a inheritance of these mutations from one individual to the next individual. So in this way we mainly study the mutation into two type. One is germinal mutation, another one is somatic mutation. Then depending upon the cause the mutations are of two types. So the first one which we studied, where we classified the mutation on the location, and the next classification of the mutation is depending upon the cause where the mutations are actually happening. So the first one is chromosomal mutation and another one is gene mutation or point mutation we can say. So let's study in detail regarding these chromosomal mutations and genetic mutation or the gene mutations. So the first one, that is chromosomal mutations. These chromosomal mutations where these are the result of the change in a number or structure of an chromosomes. So the mutations these are mainly happening due to the mutagens. The mutations are the mutation causing agents or factors. So the mutation is happening due to the mutagens which are present, or the animal which are mainly harmful are getting harmful because of these mutations. So the mutagens will be classified into physical mutations and chemical mutagens. So these physical mutations and chemical mutations where the physical mutations may be the radiations like ionizing radiations and non-ionizing radiations. So the ionizing radiations, they have a higher intensity to cause a damage in a living organisms like X-rays, alpha, beta and gamma rays. They are considered as a ionizing radiations which can harm a animal by changing their structure of the chromosome, or they may change the number of the chromosome, or they may disrupt the genetic code or a genetic makeup of an individual. Another one is chemical mutations. So some of the chemicals, they will act as a mutation causing agents. So in this way the mutation will happen in a individual. So based on a cause, we classify the mutation into chromosomal mutation and another one is genetic mutation. So the first one, the chromosomal mutations. The chromosomal mutations may be result of a change in a number or structure of the chromosomes. So the chromosome mutation will be studied based on a change in a number of chromosomes or the structure of the chromosomes. If we see a change in the number of chromosomes, that usually occur due to the errors during the meiosis or during the mitosis process. So we mainly see the instead of 46 chromosome we observe 47, or instead of 46 we observe 45 chromosomes, or we also observe the uh, addition or deletion in a chromosome number. So in this way the chromosome mutation will be seen in the change of a structure or change in a number of a chromosome. So if we see change in a number of chromosome, it usually occur due to the errors during the meiosis or during the mitosis process. These changes may involve the loss or gain of a single chromosome. That condition is called as aneuploidy. So we mainly study the chromosomal mutation where we mainly see the change in a number. Another one is change in a structure of a chromosome. If we are planning to study the chromosomal mutation where it's causing a change in a number of a chromosome, we mainly study with two type. One is euploidy, another one is aneuploidy. So aneuploidy is called as there is a loss or gain of one or more chromosomes is called as aneuploidy, whereas another type, that is euploidy, where we see the increase in an entire haploid set of chromosomes. That condition is called as euploidy. So totally we study this chromosomal mutation into two types. One is aneuploidy, another one is euploidy. So here aneuploidy, it will decrease or increase the number of the chromosome, or addition or deletion of one or more chromosome, or loss or gain of one or more chromosome is called as aneuploidy, whereas the euploidy, where there is an increase in entire haploid set of a chromosome is called as euploidy. So, next one, we study in detail regarding these change in a chromosome number. So, first one, that is euploidy. So, euploidy, where we mainly see there is a increase in a number of chromosome in a haploid set of a chromosome, or a gametes and somatic cells containing a multiples of haploid number of chromosomes are called as polyploidy. So polyploidy or euploidy is nothing but there is a complete change in a haploid set of a chromosome is called as euploidy. So there is a multiplication or duplication of the chromosome in a haploid set like 2n, 3n, 4n and 5n, 6n, etc. In the complete set, we are observing changes in a chromosomal number. So this euploidy is nothing but there is a change in a chromosome number in a complete set, or there is a change or increase in a entire haploid set of a chromosome is called as euploidy. So in this euploidy we mainly study diploid, triploids, polyploids. So polyploidy will be studied under two subheadings, that is autopolyploidy and allopolyploidy. So polyploidy, where we observe the hexaploid condition in an individual. So in an agricultural field we are implementing or inducing this polyploidy condition by treating with some of the chemical, that is colchicine or granocin. So these are some of the chemicals. So these chemicals may induce the, or they may increase the number in a haploid set. So we are inducing the polyploidy condition in a plant varieties to increase the food production or quality and quantity of the food. So we are inducing this mutation in case of the plant varieties. Then the another type of chromosomal number change, that is, or a change in chromosomal number, will be studied under another topic, that is aneuploidy. So aneuploidy, as I have already said, there may be a gain or loss of one or more chromosomes is called as aneuploidy. So the aneuploidy, in this condition, half the daughter cell produced have an extra chromosome like n plus 1 or 2n plus 1 and so on, while the other half have chromosome missing n minus 1 or 2n minus 1. So already I have said the aneuploidy, it is a loss or gain of one or more chromosome from a set of chromosome is called as aneuploidy. So the aneuploidy will be studied under two top heading, that is hyperpolyploidy and hypopolyploidy. So hyperploidy, where the number of the chromosome will be extra, or there is a gain of one extra chromosome, one or more chromosome to the haploid set. Whereas in case of the hypopolyploidy we mainly see the loss of one or loss of more than one chromosome from a set of chromosome is called as hypoploidy. So in the hyperploidy we mainly study the trisomy and tetrasomy condition. So trisomy is nothing but there is a addition of one chromosome to your set of chromosome is called as trisomy, and this trisomy will be seen in the individual which are showing Down's syndrome, or which are having a one extra chromosome on the 21st chromosome set, that is a trisomy will be seen in a 21st chromosome is called as Down syndrome, or trisomy will be seen in an individual. Then where we mainly see the 47 chromosome instead of the 46 chromosome. And another syndrome, that is Klinefelter syndrome. In this Klinefelter syndrome, where we mainly see the 47 chromosomes, where diploid set of chromosome plus one addition of an sex chromosome. So it is also a addition of one extra chromosome to the set of a chromosome is called as trisomy. The another one, that is tetrasomy. The tetrasomy, where the addition of two chromosomes will be seen to the haploid set, that is called as tetrasomy. These are the hyperploidy condition in a aneuploidy. So then hypoploidy, that is where the addition hypo means a reduction or loss of chromosome. If we are observing one chromosome is less in a set, then we can say it as monosomy. So in the individuals, we are observing some of the syndromes like Turner. So Turner syndrome, where it mainly happens in a females. So female with only one X chromosome instead of two X chromosome. There is a loss of one X chromosome will result into the hypoploidy condition in a individual. That is the female with 44 autosomes and one X chromosome, and there is a loss of one X chromosome will result into a monosomic condition in a individual. Then the another one, that is nullisomy. So nullisomy means there is a loss of two chromosome from a set of a chromosome is called as nullisomy. So nullisomy, where there is a loss of two chromosomes from a set, is called as nullisomy. So in this way we mainly study these chromosomal mutation into two types. One is based on the change in a number of chromosome, another one is change in a structure of chromosome, where in the structural changes of a chromosome we mainly study there is a loss of a fragment or segment of the chromosome is called as structural change or chromosomal aberration, we can say it. These are the changes in the number or arrangement of the genes in the chromosome and are also called chromosomal aberration. It may involve changes with single chromosome, or we can, in another words, we can say it, there is a loss of fragment or segment of a chromosome is called as structural change in a chromosomes. So the structural change can be studied through four subheading. One is deletion, another one is duplication, another one is inversion, and then the translocation. So first one is deletion. So deletion, where the chromosomal mutation, where the deletion it involves there is a loss of a region of chromosome either from the ends, or it may observe in a middle segment of the chromosome. So the deletion, it is nothing but a loss of a region or a segment from a chromosome. So the segment may get reduced from a ends of the chromosome, or it may get reduced from a internal surface of the chromosome. In this way the deletion may result into the change in the structure of the chromosome. So the deletion will be observed in a individual who are showing Cry-Do-Chart syndrome. So Cry-Do-Chart syndrome, where we mainly see the deletion of a short arm of the chromosome 5, and also in case of the Philadelphia chromosome we observe there is a loss of chromosome from the long arm of the 22nd chromosome. So in the 22nd chromosome of the long arm, where we see the deletion process, and these are the two examples for the deletion process. So deletion may alter the structure of a chromosome. So the next one, that is duplication. So duplication is nothing but a part. This is your chromosome and this is your segments. For example, for better understanding, let's make this as ABCD. And if you are observing a duplication in your chromosome, there is a multiplication or duplication of these segments will be seen in a chromosome. For example, here B part of the segment is duplicated and the rest is remained same. So in this way we mainly see this duplication process. So duplication is nothing but there is a part of the segment which is being edited or which is being duplicated, and this duplication may alter the structure of a chromosome. So the duplication, it will play a significant role in a evolution, because duplication increases the number of the genes in a chromosome complement. So in this way the duplication also result in the change of the structure of a chromosome. Then another one, that is inversion. So inversion, where we mainly see the part of the chromosome or the region of the chromosome, it will break off and it will get rotate against 180 degree, and it will rejoin the chromosome. For example, this is your chromosome and this is the segments. Here you are seeing, so this portion of the segment has been break off, then it will get rearranged as this way, A C B D E. It means the a part of the chromosome, it will rotate around 180 degree and rejoin the chromosome. So in this way the inversion also result in the structural change of a chromosome. So the inversion can be studied through two different type. One is the pericentric and paracentric. The pericentric, where the inversion or the during the 180 degree rotation, the inversion involves the centromere, then it is called as pericentric. And during the rotation the chromosome doesn't include any centromere, then it is called as paracentric. So in this way the inversion also change the structure of a chromosome. Then the last one, that is translocation. The translocation, where the region of the chromosome, it will get break off and rejoins either the other end of the same chromosome or another non-homologous chromosome. The translocation is nothing but there is a transfer of segment from one chromosome to the another chromosome, or there is a transfer of part or segment of chromosome in the same homologous chromosome. So this is your segment and this will get break off and get transferred to the non-homologous chromosome, then this is called as translocation. Or it may get transferred between the same chromosome, then this process is called as translocation. The translocation involves a region of chromosome breaking off and rejoining either the other end of the same chromosome or another non-homologous chromosome. The translocation may cause change in the morphology or appearance of the chromosome by centric fusion between the two acrocentric chromosome segments. So in this way the The structural change in the chromosome is mainly studied through these four subheading. One is deletion, another one is duplication, another one is inversion and translocation. The deletion, where the part of the segment get deleted from the chromosome and it will result into the structural change. So then the duplication, where the part or the segment of the chromosome get doubled or duplicated. So duplicated region may change the structure of a chromosome. Then the inversion, so this part of the chromosome where it will break off, then it will shift or it will rotate around 180 degree and rejoins the chromosome. So this inversion also create a structural change in a chromosome. Then the last one, that is translocation. The translocation, where the part of the segment of the chromosome, it will get transferred to the non-homologous chromosome, or it may get shifted in between the same chromosome, is called as translocation. So in this way, we mainly study these chromosomal mutation into two different types. One is based on a number of chromosome, another one is based on a structure of chromosome. In the number or a change in a chromosome number, we mainly study the Euploidy, another one is Aneuploidy. The Aneuploidy, where we mainly study hyperploidy and hypoploidy. So we should know the difference between hyperploidy and hypoploidy. Hyper means the more or addition of the chromosome. Hypo means there is a loss of one or more chromosome to the set of a chromosome. Then we studied the structural changes in a chromosome through four subheading. One is deletion, duplication, inversion and translocation. In this way, we study the chromosomal mutation. So then another one, that is genetic mutation, we study in another video. Thank you.